Sorry for the delay. Can you hear me well?
Yeah.
So, hi everyone, I'm Lucy Remark. I'm working at the Integrated BioBank of Luxembourg as an assistant scientist. And today I'm going to talk to you about induced pluripotent stem cell from a biobank perspective, from unresolved barrier to a stable production. My colleague Achilles just gave you a presentation about biobanking. I'm going to just highlight some key points. So a biobank supports research group, improve the efficiency of biospecimen storage, and conduct characterization and standard methods. Now my presentation is about IPSCs. So I'm going to review an overview of what is IPSCs. IPSCs are induced pluripotent stem cell broad program cell from somatic cell that hold unique characteristics and are able to differentiate into multiple cell types. IPSC allow to better understand the development, biology, and disease. And there are thousands of IPSCs lines established in laboratories all around the world. However, the stem cell phenotype can change by bad cell culture techniques, prolonged passaging, changing the culture condition, and therefore using bad cell would lead to a waste of time, a waste of resources, and to the generation of erroneous data which could confuse and delay the scientific progress in this area. So this is really important to have a quality control put in place, because having cell lines that have been prepared and documented appropriately, have been correctly selected and identified, and have the correct characteristic is crucial to ensure the reproducibility in IPS-based research. So I'm going to show you how we standardize in a biobank an IPS procedure and production. And all along my presentation, you are going to see this logo, the quality control check, appear at the key point. So how do we standardize IPSCs procedure and production? We start with the patient selection. We select the patient by age, by gender, by disease, mutation of interest. Then we culture the fibroblasts because we produce IPSCs from fibroblasts. I'm going to show you a case study for this. So we start by receiving the skin biopsy. We wash them and we rinse them into a drop of PBS, and we remove all of the necrotic part of our blood. We cut then the skin in 4 to 5 expand, and we place them into a flask on the endoderm side. This is really important. This is our first quality check control, because if you put it on the other side, you are going to have keratinocyte that will grow in your flask and not fibroblasts. We add the medium, we take a great care to not detach the explant, and then we put them into the incubator. The adherence of the explant is crucial. We check every week that there is no sign of contamination, the fibroblasts are growing well, and no explant are detached. If the explant is detached, we need to reattach it. Then we change the media, we test the mycoplasma to see if it's mycoplasma free, and then we do the strips initiation. All information and action are recorded on a paper form for traceability. Then we check every 2 days that there's still no sign of contamination. The fibroblasts are growing well, and the fibroblast has a good shape. If it's not the case, if the fibroblasts, instead to be elongated cells, are more like a star shell cells with filament inside, they won't be good to be turned into IPSCs. We change the media, we expand them, and at the final cryopreservation step, we do another mycoplasma test, and we stop the culture at passage 3. Then, now we have our fibroblasts ready, it's time to program them into IPSCs. To do so, we transfect and selection them. How? We put the fibroblasts and we plate them for transfection to be able to do a growth curve and to be able to do a cell pellet for fingerprinting. This is also one of our QC checkpoints. We change the media the day after, and at day 1, we measure the confluence, and we need to have a confluence of 70% for the reprogrammation. This is crucial because if it's less or more, the reprogrammation won't work and the transfection won't be efficient. At day 2, we start the puromycin selection because we only want to select the cells that have been transfected correctly. So after 8 days, we should observe in our wells that all of the cells are dead in the control well, while we have still a good amount of cells in the transfected well. Therefore, we stop the puromycin treatment when no cell remain in the control. We start to change using the medium and replace the cell on the matrigel when we have a confluence of 70–80%. This is also important because otherwise we won't see the next step coming, which is the colony emerging. You see that the cells start to group together to form like a cycle to form then a colony. You can see the colony emerging just here. And then we can continue with the colony picking, which is also a critical step. We need to cut the colony with a sterile scalpel, and we need to keep the integrity of the colonies. This is important because if the colony are destroyed, we won't have IPSCs that we will be able to expand. And one colony picked corresponds to one clone of IPS recorded as passage 0. Now, we are in the expansion phase. This phase lasts approximately 2 months. We daily exchange the media, we monitor the cells, and we do the passaging on coated matrigel plate. And then we can cryopreserve the aliquot and the cells. We cryopreserve at passage 4, 5, and 6 using a commercial solution 10% diameso base. This is also in order to standardize the cryopreservation step, and all information is recorded and archived. Now, we have IPSCs produced, but we need to test and validate the quality of the IPS produced. How? So over the expansion, we took some aliquot to do the mycoplasma test, but also the RNA extraction and the DNA extraction. Now we are able to do QRT-PCR to assess the pluripotency of the IPS by a gene analysis. We are able to also test the genomic integrity by doing karyotyping or fingerprinting, and also immunofluorescence assay for pluripotency characteristic and the 3 germ layer differentiation capacity. I'm going to show you a last example. This is pictures that we obtained in the lab. On the left side, we tested by immunofluorescence assay the pluripotency of the cells, and on the right side of the slide, you see the immunofluorescence assay done on the cells that we differentiated into endoderm, mesoderm, and ectoderm. So as a conclusion, quality checks are done at a biological level, on operational level in terms of recordings, and using a software called Laboratory Information Management System, the LIMS. The next step in the future is to automate a large-scale production of IPSCs. Thank you for your attention.
Thank you, Lucy. It was a great presentation. Now we have time for